Okay, my dear students, yesterday's life cycle of Plasmodium in mosquito diagram. Today let us see life cycle of Plasmodium or malarial parasite in female Anopheles mosquito. That is the heading of diagram. So these two essay questions are very important. Already I told you, don't ignore. As and when the topic is completed, read out. Otherwise, if you delay, you will get confusion. So what are the stages I have mentioned? You must be carefully recorded. They must be recorded in your, should be accommodated in your brain. So that's why try to practice. Sporozoite first started in the man, sporozoite converted into trophozoite, schizont, and cryptozoites, first generation merozoites in the pre-erythrocytic cycle. These stages you may remember. Then afterwards cryptozoites directly attack the RBC or may attack the liver cells against to start the exoerythrocytic cycle. In this cryptozoites also converted into trophozoite. I am recollecting the first essay question. Cryptozoites converted into trophozoite, trophozoite into enlarged cell called schizont, undergo schizogony to produce second generation merozoites. They are called metacryptozoites. They are 1000. Some are small, some are large. Larger ones are macro, they attack the liver cells, and smaller ones are the micro, they attack directly the RBC to start the endoerythrocytic cycle. In the endoerythrocytic cycle, either cryptozoite or micro-metacryptozoite, two stages start the erythrocytic cycle. So these two stages are cryptozoites or micro-metacryptozoites. They attack the RBC and convert it into trophozoite first, then signet ring stage, then amoeboid stage, then schizont. Undergo daughter cells produce, that is rosette. They are arranged like rose petals, rosette stage, and then merozoites. RBC rupture release the merozoites and hemozoin. At the time of releasing of merozoites only, hemozoin is also released. During the liberation of hemozoin, the fever symptoms appear like chill, cold. Today we discuss about the pathogenicity of malarial parasite also. You will come to know about those things. So life cycle of Plasmodium in female Anopheles mosquito diagram let us take today. Now these merozoites develop into gametocytes for further development in the mosquito, because further development of gametocytes never takes place in the human beings because of the high pH and high body temperature that is not suitable for the development of gametocytes. That's why awaiting in our blood to enter into the mosquito when the mosquito sucks the patient's blood. So these gametocytes enter into within a week, they must enter into the mosquito, otherwise they regenerate, they die. So these gametocytes are of two types according to size. Smaller ones are the micro, larger ones are the macro. Micro also called male, macro also female. And micro gametocytes undergo gametogeny to produce the male gametes or sperms 8, and female gametocyte undergo maturation to produce egg. So at the fertilization cone, the fertilization occurs. So after fertilization, union of pronuclei leads to formation of synchorion, and that egg with synchorion is called zygote. Zygote elongates and becomes slender and moving stage called ookinete or vermicule. It penetrates the stomach wall and settles below the basement membrane. Already told you, epithelial cells are arranged on basement membrane. Below the basement membrane, these stages are settled and form a cyst around it called oocyst or sporont. Undergo sporogony to produce number of daughter cells, mainly 1000. So with all these 1000 nuclei are surrounded by inconspicuous settlement of cytoplasm, causes the sporoblasts, development of sporoblasts. These sporoblasts undergo repeated mitodivisions to produce 10,000 sickle-shaped cells called sporozoites. Now the cyst with sporozoites is called sporocyst. Sporocyst ruptures and releases the sporozoites, which enter into the salivary glands where they are stored for the time being. And the storage of sporozoites occur in the female Anopheles mosquito, where that place is salivary glands. You must remember this is very important neat bit also. Yesterday I told you about this. So now let us take this sporozoites. When the same infected mosquito bites the healthy human beings, then these sporozoites injected into the blood circulation. They stay for the period of 30 minutes, and after 30 minutes they attack the liver cells. Thus again cycle is continued. Again life cycle in man. Again mosquito. So like that. Now you take this diagram. Two essay questions. Briefly I have recollected those essay questions. Now let us take the structure and diagram of the life cycle of Plasmodium in mosquito. This is the crop of the mosquito. We have to draw the crop of the mosquito first of all. And crop wall this is. This is actually crop wall. And this is lumen of the crop. Already I told you the fertilization, everything occurs in the lumen of the crop. You say crop, not stomach. In some areas it is used stomach, but it is not correct. Everything occurs in the crop. And then two types of gametocytes enter into the. So these are the small micro gametocyte and large macro gametocyte. And then they enter into this micro gametocyte and macro gametocyte. And this micro gametocyte divides with nuclei also. We must mention nuclei also here. The large nucleus in the micro gametocyte and small nucleus in the macro. This nucleus again draw here, and now it is divided into number of nuclear bits. These nuclear bits are nearly how many? Male gametogeny 6 to 8 microgametic nuclei are formed. So around each nuclear bit, the cytoplasm is surrounding the each nucleus like a flagellar structures. These are cytoplasmic flagellar structures. These cytoplasmic flagellar structures exhibit lashing movements to release the gametes. Thus the gametes are released. Microgametes are released. And coming to this, put an arrow mark here. Here also arrow mark. And in this case, what happens, here the microgametocyte, to some extent macrogametocyte enlarges and form a fertilization cone. Inside the fertilization cone, this macrogamete nucleus is actually reached the periphery. This is periphery, and all this is some cytoplasm comes out as a projection. This is called fertilization cone. This projected part is called fertilization cone. So now the egg is produced. So this is, now we call it egg or female gamete. This is macrogametocyte. The microgametocyte. Of course here labeling towards right side is not possible. Always when you draw any diagram, not only this, any diagram, you have to keep labeling right side of the diagram. Always, this is best precautionary measure. But in this case it is not possible. Both the sides we have to draw the diagrams and labeling also. So this is here also you can write down female gametocyte. Female gametocyte or macrogametocyte, and male gametocyte or microgametocyte. These are coming from when the mosquito sucks the patient's blood. These enter into the lumen of the crop of the female Anopheles mosquito. This is male gametocyte, or micro or male, and macro or female. In the bracket you write down here also female gametocyte. Okay. Now formation of gametes is over. These are the male gametes or sperms. The bracket you write down 6 to 8 male gametes are produced. Minimum 6, maximum 8. And one of this male gamete comes, and here at the fertilization cone it tries to enter. At the fertilization cone. Here it tries to enter. Here there is a nucleus. So both the nuclei united. Fertilization takes place. This is the site of fertilization. Both male and female gametes are going to union. Fertilization, type of fertilization in Plasmodium vivax is anisogamy because of the union of dissimilar gametes. and then form into a zygote, a round structure. This zygote is produced. So this is zygote. This stage is called zygote. Zygote in which the nucleus is present. This nucleus is called syncyrion. So maximum towards right side we have completed the labeling. Now left side we are coming. This is the wall of the stomach. Wall of the stomach is richly provided with epithelial cells. All these are columnar epithelial cells. Any gut wall is made up of columnar epithelial cells. So these are columnar epithelial cells. So this zygote enlarges gradually. You see here, this zygote is elongated and moving like this, penetrating the wall of the stomach. This moving zygote is called wukinet. Wukinet also called, other name of wukinet, vermicule. So for the second life cycle, you have to draw this diagram. All these stages occur in the crop of the stomach, in the lumen of the crop of the stomach and on the wall of the crop of the stomach. This is the wall of the crop. This is lumen of the crop. Already it is mentioned here, lumen of the crop. Now this wukinet attains a round shape with a zygotic nucleus or syncyrion. So around which the cyst formation occurs. The cyst is also formed. Now you call this stage as oocyst. Already I told you what is the other name of oocyst? Sporant. This sporant undergo sporogamy. So outside the wall of the stomach and below the basement membrane, all these stages are going on. So here sexual cycle is completed in the lumen. In the lumen of the crop, sexual cycle is completed. You can mention in the bracket, sexual cycle is completed. Now outside the lumen means on the wall of the crop, it is called sporant, oocyst. Sporant undergoes sporogamy. So sporogamy occurs on the wall of the crop. Gametogamy occurs in the lumen of the crop. Okay. Now this oocyst, in which the nucleus divides into number of nuclear bits. So this is oocyst. Number of nuclear bits. Number of oocysts also let us draw here like this. So because all these stages we have to draw, that's why. Now this nucleus divides by reduction division followed by mitotic divisions with many daughter nuclei. Each daughter nucleus is surrounded by almost a bit of cytoplasm like this. So that's why these are looking like inconspicuous cells. This inconspicuous cells cellular stage is called sporoblast. Now in the sporoblast, repeated mitotic divisions occur. So due to the repeated mitotic divisions in the sporoblast, the number of daughter cells are increased to 10,000. Daughter nuclei are produced. All these 10,000 daughter nuclei are surrounded by, here these are all daughter nuclei, surrounded by what you know? Cytoplasmic bit like this, which are sickle shape. Like sickle shaped cells are produced. So around which still cyst is there. Still the cyst wall is there. So sporozoites with cyst called, these are all sporozoites. Sporozoites with cyst is known as sporocyst. This cyst is called sporocyst. Don't say oocyst. Oocyst at this stage only it is oocyst, where the zygote is present. Now this is sporocyst. Now this sporocyst ruptures and bursts. It ruptures and bursts to release these sporozoites. Again we have to draw these sporozoites. These sporozoites come out. Sickle shaped sporozoites are coming out and settled in the salivary glands. These are all sporozoites which are sickle shaped. So all these are sporozoites. Now ready to enter into man, a healthy man. This diagram you have to draw. Of course, as far as diagram is concerned, we may feel some difficultiness. I can assume it, but we have to try to maximum practice for these two diagrams, life cycle of Plasmodium in man as well as yesterday's diagram. Remember yesterday's diagram? I can go back. Now you see that diagram also. Here you see. Man, Bell's mosquito. Now this diagram. You see this diagram. Pre-erythrocytic cycle, exoerythrocytic cycle diagram. This one. This is pre-erythrocytic. This is exo, and then micro metacryptomyzoids attack the RBC. All these brown colored, brick red colored cells are RBC, in which the parrot green colored stages are there. All stages of the Plasmodium in the RBC: trophozoite to signet ring, amoeboid, schizon to rosette, merozoites. So this diagram for life cycle of Plasmodium in man, asexual cycle, and this now whatever the diagram that is drawn by me is the life cycle of Plasmodium in mosquito. So this diagram. This is also called sexual cycle. In the bracket you write down sexual cycle of Plasmodium, or Ronald Ross cycle. This is also called Ronald Ross discovered the oocysts on the wall of the crop. Hence this is also called Ross cycle. That is Golgi cycle. This is Ross cycle. So with this, the second 8 marks question we have completed. Now let us pass on to the next page. Pathogenicity of malarial parasite. Pathogenicity means disease symptoms. How it affect the human beings. Let us see. Pathogenicity means disease symptoms. One by one, let us list out the disease symptoms. So the malaria fever appears in 3 stages. The fever caused by Plasmodium is called malaria fever. Okay. This malaria fever appears in 3 stages. These 3 stages separately we can discuss. The first one is cold stage. In the cold stage, 3 symptoms appear. Cold stage itself, that name is there, which is nothing but chill. Chill means shivering. Chilled fever occurs means fever with shivering. Gaza gaza unikinchestadi annamata. So you cover with rugs or kambali like that, it's of no use. Such a shivering occurs. So chill, cold stage chill, and chill, headache. Symptoms carefully write down. Giddiness. These are the 3 symptoms of cold stage. Giddiness. Okay. Now hot stage. Hot stage means temperature. Here also symptoms are there. What are the symptoms? High temperature, maximum 104 degrees centigrade, 102 to 104 degrees Fahrenheit temperature. Hot stage, by name itself it is there. Hot means temperature stage. High fever, 102 to 104 degrees Fahrenheit. Breathe rate and pulse rate increases. Breathe rate as well as and pulse rate increases. This is another symptom, very dangerous at this time. Tiredness also there. Then sweat stage, cold, hot and sweat stage. 3 stages. Malaria fever can be discussed under the 3 stages. So profuse sweating occurs after temperature. Immediately temperature subsides. Profuse sweating occurs. And low temperature. Temperature is reduced totally, almost reduced. profuse sweating followed by low temperature, or low temperature followed by profuse sweating. చల్లగా అయిపోద్ది, మళ్ళంతా కూల్ అయిపోద్ది. Again మళ్ళీ cold stage shivering, and again hot stage temperature fever, and again sweat. These three are repeatedly occurs every 48 hours. It repeats. That's why this malaria fever is called benign tertian malaria. Plasmodium vivax causes benign tertian malaria. Already 4 types of malaria fevers names I have mentioned: malignant tertian, benign tertian, mild tertian, and quartan malaria. So this is as far as pathogenicity of malaria is concerned. So besides this, the another symptoms also occur because of the destruction of more RBC. Anemia occurs because majority of the RBC are destroyed, because while releasing merozoites, RBC get ruptured. Thus so many RBC will be destroyed. So anemia, that leads to anemia. And tropical splenomegaly syndrome. Tropical, this is the first symptom, malaria fever. Second symptom is this one, tropical splenomegaly syndrome, because this is a tropical malaria. That's why splenomegaly means enlargement of spleen. Megaly, mega means large size. Splenomegaly syndrome also appears. What is the meaning of splenomegaly? You know, enlargement of spleen due to the over immune response to the fever given by spleen. Spleen is one of the most important lymphoid organ. Bone, spleen, bone marrow, lymphoid organs, all these are lymph nodes. They are all lymphoid organs. So the spleen gives exaggerated immune response, over immune response. That over immune response leads to the enlargement of spleen. That's why that condition refers to splenomegaly syndrome. So anemia and tropical splenomegaly syndrome caused due to immune response given by spleen. Due to immune response by spleen. That is. And another last and final symptom, already suffered from malaria. A patient already suffered from malaria, but now sometimes this relapse of malaria also caused. Means relapse of malaria means again the malaria attacks the same person, already suffered, but again he will be attacked. So that is called relapse of malaria. If malaria is re-attacked, then it is called relapse of malaria. How it occurs? You know, some of the macrometacryptozoites enter into liver cells. See, metacryptozoites, how many? 2 types. Macrometacryptozoites are larger one, micrometacryptozoites are smaller one. The smaller micrometacryptozoites attack the RBC. Larger macrometacryptozoids enter into the liver cells and take rest. So some of the macrometacryptozoids, how the relapsing fever occurs, let us see. Some of the macrometacryptozoids enter the liver cells to undergo rest, to undergo dormant condition. Dormant means rest. To undergo dormant condition. The bracket you want, rest. So these are called hypnozoids. These macrometacryptozoids are hypnozoids. These are also called, hypno means rest, sleep, means dormancy. Hence they are called hypnozoids. Hypnozoids should be underlined. So when these hypnozoids are activated, that is a problem. Sometimes these are activated. Already in the inactive state they are in the liver cells. When they are activated, when these are activated, this clause must be there. When these are activated, what happens? You know, They attack the RBC again. They attack the RBC to produce merozoites. This is called relapse. Attack the RBC to produce merozoites, because erythrocytic cycle end products are merozoites. Last third generation merozoites are simply merozoites. Again fever symptoms appear. This is called relapse of malaria. Again fever symptoms appear, because RBC ruptured and released the merozoites and hemozoin. Hence, during liberation of hemozoin, always fever symptoms appear, called relapsin or relapsing of malaria. Relapsing of malaria. తిరగబెట్టిన మలేరియా జ్వరము అని అంటాం మనం తెలుగులో. తిరగబెడుతున్నా అంటే తిరిగి వస్తున్నా, అగైన్ ఇట్ ఈజ్ అటాకింగ్ అని. దట్స్ వై ఇట్ ఈజ్ రిలాప్స్ ఆఫ్ malaria. సో లైక్ దట్ pathogenicity can be discussed under the 3 headings: malaria fever, benign tertiary malaria, symptoms 3 stages: cold stage, hot stage and sweat stage, and anemia and tropical splenomegaly syndrome and relapse of malaria. So that's now control. How can we control malaria? Let us take this. Control means prophylaxis. Prevention is better than cure, you know. That slogan we should not forget regarding pathology. Pathology means study of diseases caused by pathogens. So regarding control of pathogens, we have to always take care of not attacking to the human society especially. That is prophylaxis or prevention, control. So how can we prevent? Prevention, or you can also say control. Already malaria fever attacked, then you have to take the drug quinine. Quinine is a malarial drug, anti-malarial drug. In the bracket you write down, anti-malarial drug is a quinine. It is an alkaloid. An alkaloid obtained from bark of the Cinchona officinalis. All alkaloids help in curing so many disorders in the human beings. So quinine is a anti-malarial drug, an alkaloid obtained from bark of Cinchona officinalis. Bark means outer thick layered structure of the trunks is called bark. From the bark of Cinchona officinalis, the botanical name of this plant is Cinchona officinalis. This is malarial plant also, people say generally. Underline this. Bark. This drug is obtained from which part of the plant? Bark. Quinine is a most important malarial drug. Kills the erythrocytic merozoites. When the erythrocytic merozoites are killed, disease symptoms will not appear. So that is. And second control is chemical control. Chemical control involves what you know. This is medicinal control. First one. Chemical control involves the mosquito repellents. You know, mosquito repellents means good night, jet, all out, tortoise coils. They are all we use, you know. And mosquito nets, mosquito repellents and nets. Nets also, mosquito nets also we use. Of course, mosquito nets that much not curable, but mosquito repellents to some extent, because all the mosquitoes will not die, but will be senseless at least for few hours, not total night, for few hours. We can protect ourselves from the mosquito bite at least that 2 to 3 hours. So chemical control involves the mosquito repellents like goodnight, jet, all out, Odomas. So many are there, tortoise coils, all these are. And if you can use pyrethrum oil, this is very important oil. Pyrethrum oil and kerosene can be sprayed on the breeding areas. Kerosene can be sprayed. స్ప్రే చేస్తే తగ్గిపోతాయి. స్ప్రేడ్ ఆన్ ఇన్ ది their dwelling areas, breeding places where the eggs are deposited. Generally mosquitoes lay eggs in the water, surface of water, so that is the breeding area. Sprayed in breeding areas, so that at the level of larva itself they will be killed. Life cycle of mosquito stopped at the larval stage itself. Larva becomes pupa, egg larva, pupa, imago are the 4 stages in the life cycle of mosquito. So these larval forms are wrigglers. So at least at the stage of wrigglers they will not get oxygen supply to the oxygen supply, because of the entire surface of the water is occupied by this oily layer. These 2 oils, pyrethrum oil is very important, kerosene also, they occupy the surface of the water, so that the atmospheric oxygen cannot be dissolved in the water. It will be not in the vicinity of larval forms and they die of suffocation. That is best remedy out of this chemical control, mosquito repellents, nets, etc., DDT. And also you can use DDT and Gamaxin, spraying of DDT and Gamaxin. DDT, Gamaxin, these are all powerful insecticides, Gamaxin, and BHC, benzyl hexachlorine. DDT means dichloro-diphenyl trichloroethane. BHC means benzyl hexachlorine. And Gamaxin generally. Any important leaders, political leaders or ministers visit the cities, municipalities, they will spray this DDT powder or Gamaxin powder or BHC powder on the side of the roads. You can see at that time only you will see. The other in the other period, normal days, they won't care. But this is also not powerful insecticides. They also never kill the insects. They develop immunity. Insects develop immunity against these insecticides, DDT, Gamaxin and BHC. So these are all chemical control. And most important control out of all is biological control. Of course these headings are not there, but I am keeping these headings. Control is separated under 3 headings. Medical control is the first one, chemical control second one, last important control, biological control. So as far as biological control is concerned, the Gambusia, a larvivorous fish, is introduced. Most important fish, a larvivorous fish. It eats away the regular larva. Larvivorous fish introduced in the drainages where the dwelling areas of mosquitoes as well as egg deposition area also. So introduced ovipositing area. Oviposition means egg deposition. Introduced in the drainages. So helps a lot to control the population of mosquitoes at the time of breeding stage itself. And also another one is there. It eats away the larvae of the mosquito. Larvae of the mosquito regular. And Utricularia, bladderwort, a trap leaves. In the botany also you can come to know about these things. What are they? Insectivorous plants. Utricularia, Nepenthes, Drosera, all these are pitcher plants, Nepenthes. They trap the insects. That's why those leaves are called trap leaves. So Utricularia, common name is bladderwort. It has valves. So those valves are opened to attract the insects. And then whenever the insect enter into the Utricularia, the valves are closed and digest. So like that they trap the insects. Utricularia also trap the insects and kill the insects, digest the insects. Best Gambusia and Utricularia, if you introduce in the drainage systems, definitely the mosquito breeding phenomena or process is obstructed. Like that the mosquito population is controlled. So if you control the mosquito population, we can prevent the mosquito bite. If you can prevent the mosquito bite, entry of sporozoite also stopped. Thus we can be free from this malarial attacks. This is about the prophylaxis. So pathogenicity 4 marks question. You take the 4 marks question of this. Now let us take the next life cycle of biology and human welfare. So here 3 essay questions we have completed. What are those 3 essay questions? The first essay question is life cycle of Entamoeba histolytica. Second essay question is structure and life cycle of Entamoeba. Second, life cycle of Plasmodium in mosquito, third life cycle of Plasmodium in man, and fourth essay question life cycle of Ascaris lumbricoides. So this is the Ascaris lumbricoides, a first nematode parasite. So we are going to complete the syllabus almost very fastly. At the same time, we are giving the information slowly. So both are covering of syllabus is going on, and explanation is also going on very slowly. Of course, anyhow, if you come tomorrow for offline classes, I think it is so happens tomorrow you will come. I don't know whether you come. Is it correct, girls and okay, all of you? Can you give me information? Of course, we didn't receive any message. Cancellation message only we received, and again when you come, we don't know. Prabhu?
Sir, I think I am not coming, sir, because I am stuck with it.
Oh, you have not
So
also.
I opted online, sir.
You have also not received any message.
Yes, sir, we received message, sir.
Uh,
I
to,
was offline and both online, sir.
okay. So tomorrow offline is there or not?
There, sir, it's
Uh,
there, sir.
okay. Right. Tomorrow we can meet. Okay. So anyhow, next is a question. Ascaris lumbricoides, 8 marks question. Another 8 marks question. Structure and life cycle, very simple question. If it comes, you can easily answer this essay question. Ascaris lumbricoides is a nematode parasite. So let us take the classification. Phylum Nematoda or Ascalmenthes or Nematihelminthes, or simply you can say Nematoda. Class Phasmidia. Only these two you mention. This is the systematic position of the Ascaris lumbricoides. It belongs to phylum Nematoda and class Phasmidia. So already in the Nematihelminthes, two classes we have mentioned, Phasmidia and Phasmidia already I have mentioned. So you can know about this. And let us take the introduction of Ascaris lumbricoides. Then structure. First introduction. It is a tropical parasite, mainly tropical parasite, because tropical area is more in the world than other areas. Cosmopolitan in distribution. Cosmopolitan means worldwide. Cosmopolitan in distribution means worldwide in distribution. And monogenetic parasite. This is it, like Entamoeba histolytica. It completes its life cycle in only one host, that is man. Primary host is there, secondary host is absent for the Ascaris lumbricoides. Monogenetic nematode parasite, if you are asked, your answer must be Ascaris lumbricoides. This is sexually dimorphic. Male and female individuals are separate. Means sexes are separate. That's why it is a dimorphic parasite. Monogenic dimorphic parasite, and it is also coelozoic parasite because it lives in the lumina of the small intestine. Lumina of the small intestine, coelozoic parasite. Large intestine parasite is Entamoeba. Small intestine parasite is Ascaris. These two are the intestinal parasites we have studied. So that is why Ascaris lumbricoides is also called enterozoic parasite. Is also called enterozoic parasite. Of course, time is very short. Only 5 minutes is left.
Sir, what is dimorphic, sir?
Dimorphic means, di means two, morphe means two forms. It is in male and female. Males and females are separately found in the Ascaris. Not only Ascaris, all nematodes are dimorphic. In the general characters of nematodes, just you refer, if you refer the general characters of nematoda, you will find this point, sexual dimorphism. These are dimorphic sexually. If you want, you can add sexually also. Male sex and female sex are separate, hence sexual dimorphism is noticed here first time. And coelozoic, enterozoic parasites, and this is a common roundworm, most common roundworm. of all roundworms, this is the most common roundworm and common enteric parasite. Enteric means gut parasite. That's why it is common enteric parasite also. Common enteric parasite and common gut parasite. Most common enteric parasite, Ascaris lumbricoides, from children to adults in the intestine, because those people who purchase the sweets and all these things outside and eat away, definitely they will get this roundworm. So most common, that is why common enteric parasite, monogenetic parasite, dimorphic parasite, sexually dimorphic parasite, coelozoic parasite, and pseudocoelomate. You all know pseudocoelom, a body cavity without lined by mesoderm. That's why these are the pseudocoelomates. So monogenetic, dimorphic, pseudocoelomate parasite, these three clubbed together. That question they ask like that: monogenetic, dimorphic, sexually dimorphic, and pseudocoelomate parasite. That is Ascaris lumbricoides. Okay, so these are the most important introductory features. Lumbricoides, why the name is given lumbricoides? The Ascaris means elongated. Lumbricoides means it resembles earthworm lumbricus. It resembles European earthworm. In the earthworms, I have mentioned this British earthworm, that lumbricus. So that's why the species name given lumbricoid. It resembles the earthworm lumbricus. Earthworm lumbricus. That's why the name lumbricoid is. So this is Ascaris lumbricoides introduction. Now let us take the structure of Ascaris. Tomorrow life cycle we can take. Today's structure. Male and females are elongated. Both male and females are elongated. Are elongated. They are cylindrical in shape. They have mouth in the anterior end. Mouth in the anterior end. I will draw the diagrams, both male and female. You can see that. Mouth in the anterior end. Mouth provided by, mouth surrounded by or provided by 3 chitinous lips. This is very, very important point. You put a star mark. Neat also important. 3 chitinous lips. Lips made up of chitin. Hence 3 chitinous lips are present. And just below the mouth, excretory opening is present. Just below the mouth. This is the common resemblances of both male and females. Below the mouth, excretory pore is present. Lies the excretory pore. Excretory pore lies. Now, how can you distinguish the males from females? So, male Ascaris and female Ascaris. Let us distinguish between those two. This is also important for 2 marks question. Distinguish between male and female roundworms. That question is there in the question bank. Male Ascaris and female Ascaris. First difference, curved posterior end. Curved posterior end. Here, hook-like, curved hook-like posterior end, straight posterior end. Straight posterior end. This is a major difference. In the curvature, in the posterior side, cloaca opening is present. Cloaca lies in the curve of posterior region. In the curve of posterior end. Here, this is a almost tail. Posterior end means tail. And here anus lies at the posterior end. And 2 equal cupulatory spicules are present. 2 equal penial setae are, they help in the transfer of sperms into the females. 2 equal penial setae / cupulatory spicules are penial spicules present. Here they are absent. In the females they are not, but in the females, female genital opening or female reproductive opening lies 1/3 from the mouth, in the anterior end, lies 1/3 from the mouth. But here male reproductive opening present in cloaca, present in cloaca. These are the differences, four differences. So you have to write down like this. This is the structural features of the Ascaris lumbricoides. Now let us take the Ascaris lumbricoides structure. This is the male with curved hook-like posterior end, and this is the female with straight posterior end. Here the cloaca is present with pair of cupulatory spicules, mouth is present, both excretory pore is present, genital opening is present. This is anus. This is anus. This is female reproductive opening. This is excretory pore in both the cases. This is mouth. And this is cloaca, because this is male. And cupulatory spicules or pineal spicules. So thus we can distinguish the males from females. And also one more difference is there. Males are shorter than females. Males are shorter and females are longer. This is the major difference, externally visible difference. Male Ascaris, this one male Ascaris, and this is female Ascaris. Thus we can distinguish the males from females. Tomorrow we can see the life cycle. Okay, I can stop the class.
Thank you, sir.
Okay, bye.
Thank you, sir.
Bye, Amma.